Hello and welcome to installation number 2 in our chapter on DNA. In this section we're going to be looking at how DNA is actually replicated. Now, most of our discussion this semester related to the idea of structure and function and their interconnectedness has related to DNA. Has related to proteins, but if on the AP exam you're asked about other examples of relationships between structure and function, DNA replication is a great example because the structure of the DNA molecule with the bases on the interior held together by hydrogen bonds is strongly suggestive of how that molecule is actually able to make copies of itself. And DNA is replicated through a process called semi-conservative replication. Now, semi, again, meaning half. Conserve means you're saving. So in essence, when you split this apart, you don't just get rid of the parental DNA. You actually use the parental DNA to synthesize or create the new strand. So in essence, when they talk about daughter DNA molecules, each daughter molecule that's made has one older strand or parent strand and then a newly synthesized strand. This is sort of easy to visualize here. We see the original DNA or parental DNA. Now, an enzyme will break the hydrogen bonds between these bases. Then another enzyme can go in and add bases to the original strand that are complementary. So you wind up with two daughter molecules, two daughter DNA molecules, one with an existing parental strand or original strand, and then the other strand being new. Now, when we look at the overall chromosomes and their replication, we notice a couple of significant features. One is that, this may be good to start down here. If we look at the two strands of DNA that we see in our chromosomes, we have these long linear chromosomes, these long lines. We have two original strands. And what will happen is bubbles will actually open up. An enzyme will unzip these strands so that they form these little bubbles. Okay. Now, these bubbles are referred to as the origin of replication. So, they'll mention this here, the origin of replication. Now, one difference between bacteria and eukaryotic cells is that bacteria will only have one origin of replication, but in eukaryotic cells, these long linear chromosomes, you can have hundreds, maybe thousands of origins of replication. So you can start separating the DNA and copying it in many locations along a chromosome, and that helps an individual copy that information more quickly. Additionally, when these bubbles open up, the replication or the newly synthesized strands go in both directions. So the bubbles increase in size until eventually they're all connected, and you wind up with two new strands. Again, one original and then one new. Let's see. Again, in terms of terminology, origins of replication are in essence these bubbles, and that's where we see the replication begin. Again, in our eukaryotic chromosomes, we see hundreds, potentially thousands of these little bubbles where replication begins, and again, this process occurs in both directions. Now, this is a very helpful diagram because it helps sort of visualize what we're going to talk about here shortly. We know that the order is in the bases, or the code is stored in these bases, these A's, T's, C's, and G's. But to get at the code, you need to separate the strands. So this enzyme, helicase, plays the role of the enzyme that separates the strands. So ACE tells us that it's an enzyme. Helicase suggests that it plays a role in breaking down this double helix. So helicase will locate a specific sequence along the DNA molecule and then attach at that location and then begin sliding, or the molecule begins sliding past the helicase. When that happens, it separates these strands. Now, if you've ever taken a string or a piece of yarn and try and pull those strands apart, you'll notice that there tends to be kinks that are made in the yarn, and that stops up that separation. Well, the same phenomenon can occur in DNA. So there's another enzyme called topoisomerase that actually cuts the DNA so that it can swivel or turn and unwind and relieve some of that pressure. So helicase is important because it unzips the DNA. Topoisomerase is important because it cuts and relieves the tension on the DNA. Let's see. We also know that if left to itself, DNA would have a tendency for these nitrogenous bases to reform their hydrogen bonds. So there's another protein or complex of proteins called single-stranded binding proteins that actually prevent the nitrogenous bases from forming hydrogen bonds yet again. Let's see. And primase, we'll talk about its significance here in just a little bit. Now, in terms of terminology, when you create this origin of replication or this replication bubble, the replication fork is this region here where the split is occurring. So where the helicase is acting is this Y-shaped region where the information is being added. Again, helicase is that enzyme that splits apart the strands. Single-stranded binding proteins, again, attach to the parental strands and keep them separated from one another so they don't reform bonds. And topoisomerases, again, cut and swivel this DNA so that it can relieve some of the tension that's placed on the molecule as it opens up and unzips. Now, DNA polymerase is the key enzyme here. It's an enzinase, so we know it's an enzyme, DNA polymer. So what this enzyme does is it adds nucleotides. So it adds the sugar phosphate nitrogenous base to have the A's, T's, C's, and G's. So say for instance, this molecule has got an adenine. DNA polymerase will add in a nitrogenous base that has a thymine. Or if this is C, DNA polymerase will add in another base that has the G. So DNA polymerase is the key enzyme that attaches new bases to the newly synthesized molecule. So DNA does the assembly of the new strand of the DNA molecule. Now there is a key shortcoming to DNA polymerase. It can only add bases to the 3' end of the existing strand. Now, that's significant because what that does is it limits DNA polymerase to only being able to add DNA to where strands already exist. It cannot create a brand new strand. The creation of a brand new strand actually falls in the shoulders of this RNA primer. Now, what happens is before you even start synthesizing a new DNA strand, you actually get this RNA lay down a short sequence so that DNA polymerase can attach to it and begin adding DNA nucleotides. So this image helps us sort of visualize what's going on here. So we can look at this, and here you see DNA synthesis occurring in the 5' to 3' direction. So the molecules are being added to the 3' end of the existing strand. But again, DNA polymerase needs to attach its nucleotides to an existing molecule. So RNA polymerase lays down an initial sequence called an RNA primer or RNA primer, depending on how you pronounce that. But it's just a short sequence of a nucleic acid, or a ribonucleic acid, or a short sequence of RNA. Once that occurs, then DNA polymerase can attach to that 3' carbon and then begin adding molecules of the DNA nucleotide, or DNA nucleotides, to make the DNA polymer. So again, primase is this enzyme that starts the whole process. It lays down the initial nucleotide, so then after that short primer or primer is created, only 5 to 10 bases long, the new DNA strand can begin to be synthesized. Okay. Now, again, DNA polymerase is going to add those bases after the RNA primer or RNA primer. So here we have the RNA sequence that's laid down. DNA polymerase comes in, and then it will add A, T, C, or G nucleotides as needed. So then you start to synthesize this new strand. So for DNA polymerase to work, again, it requires this primer or primer, and it requires this template DNA so it can match up nucleotides with their appropriate partner. And sometimes people ask how quickly this occurs. About 50 bases per second, so pretty remarkable rate. So this image is also nice because here you see helicase breaking the strands, breaking the hydrogen bonds between the bases. Here you see the primase laying down that initial RNA sequence that will end with the 3' carbon being exposed. Then DNA polymerase can link in and start bringing in bases that are floating around inside the nucleus and then attach them at the appropriate location. Sweet. This is another view of that process. What we see is DNA. Remember the numbering of the carbons. This asymmetric carbon is 1, 2, 3, 4, and this is the 5th carbon here. So again, as the strand is being synthesized, you can only add new bases to the 3' end. Okay, so it has to be the 3' end. Oops, I'm sorry, the 3' end to which the new bases attach. Now, we call these nucleotides, but what actually gets added is a structure called a nucleoside, since it has these 3 phosphates. Well, what DNA polymerase does is when it joins up these molecules, is it will actually split off these 2 phosphate groups to form what's called a pyrophosphate molecule, these inorganic phosphates. But the basic idea is you have these nucleotides, or more precisely nucleosides, floating around inside the nucleus. DNA polymerase helps link them up, and it breaks off these phosphate groups so that you can form a brand new nucleotide in the DNA molecule. Again, only adding to the 3' end. Let's see, again, nucleoside triphosphate, you may see this listed as DATP, is what gets added in. So all they're saying is you take this molecule, split off these phosphates, and then DNA polymerase links it up with the existing chain to the 3' end of the carbon. All right, we'll pause there.